There was a specific day my dad brought this particular wine home. Although we cannot really remember the name of the wine, but it was a very brilliant wine. And I was lost in thought. How was such a brilliant wine produced? Or a few of us might love different beverages like vodka, Smirnoff, and so forth. I may also be lost in thoughts, wondering how such a drink was produced. Well, in this video, we will discuss the biochemistry behind wine and liquor production. The biochemistry behind wine and liquor production is alcohol fermentation. But first, let us understand what alcohol fermentation is. What is alcohol and what is fermentation? Okay, what are alcohols? If you remember in our chemistry classes, we said that alcohol refers to a class of organic compounds containing a hydroxyl group that is attached to a carbon atom. Some common alcohol include ethanol, methanol, and propanol. Now, alcohol fermentation, which is also known as ethanol fermentation, is a biological process where sugar like glucose, fructose, and sucrose are converted into ethanol and carbon dioxide by yeast, bacteria, or other microorganisms. Remember in our previous video, we said that fermentation occurs in the absence of oxygen and the electron transport chain to produce ATP. Now, the sugar is being converted to ethanol and carbon dioxide by yeast fungi and mostly Saccharomyces cerevisiae to produce either wine, beer, and also remember that the CO2 that is produced is also what is used in bread making that makes the bread rise. Is because CO2 is trying to escape from the bread and which makes the bread to rise, to be puffy. Let us talk about the process of alcohol fermentation. Now, alcohol fermentation involves various steps. One is glycolysis. Now, glycolysis is the initial stage of alcohol fermentation, where glucose, which is a 6-carbon molecule, is broken down into pyruvate, which is a 3-carbon molecule. Now, to the formation of pyruvate, 2 ATP is also formed and 2 NADH is formed. Now, the carboxyl group of pyruvate is removed, releasing carbon dioxide and forming acetaldehyde. But it did not stop there because we need to regenerate NAD+, and that was the key point of fermentation, the regeneration of NAD+. Now, for NAD+ to be regenerated, 2 hydrogen molecules of the NADH is being oxidized to form ethanol. Now, when ethanol is formed, that 2 hydrogen molecule that was oxidized now leads to the formation of NAD+. Now, NAD+ is now regenerated. Now, glycolysis again cannot take place. And that is how the cycle continues. Application of alcohol fermentation. Alcohol fermentation is applied in beer making, wine making, alcohol making, the making of spirits, which is used to dress wounds, and it's also applied in food preservation. Also applied in the baking of bread, that is the carbon dioxide that is trying to escape from the bread is what makes the bread puff up. And alcohol fermentation is also applied in biofuels. In our next video, we'll talk about lactic acid fermentation. If this video have helped you, please like, share, subscribe, and also tell your friends concerning the Biochemistry Academy.